Hello friends, welcome to lecture 3 of week 4. So in the past lectures we have seen the basics of genomics and transcriptomics, what is the need, why we should do, how to plan the studies. And in this one we will look at the workflow that we use for carrying out the experimental part, okay, and which is important to understand because when you analyze the data you should know that what experimental protocol has been followed, okay. So basically what has happened recently is that the cost of sequencing and even the the amount of data that is generated by these methods is increasing every day, okay. And because of this it is becoming more affordable, and so many as you can see that basically there is a huge decrease in the cost, and in terms of the data generated you can say there is a huge increase, okay, in terabytes. So basically the reduced cost and affordability and accuracy is luring users to generate more and more data. And as you can also check, for example, in case of published plant genomes, just for 2020 if we see, the number of genomes at chromosomal levels have been increasing exponentially, and at the non-chromosomal level also. So we will discuss what is chromosomal and non-chromosomal later in this series of lectures. But okay, so this way more people are able to do or handle complex genomes, okay. And so basically in this slide I am showing you the overview that we will also see in detail further. So it all starts with the sample collection. So this species that you are interested in, appropriate sample of that species has to be collected. It should be appropriate. So if this species is available for you, like it is a plant that is growing in your backyard or in your campus or a nursery, okay, then basically okay, you can still like continue taking the samples later also. But sometimes the samples are only one time, that it is seasonal, or basically you have got from an animal or bird where ethics and those things are involved, and you cannot always take out blood or take another tissue from using any invasive method. So you should have sufficient amount of the sample collected, okay, for your purpose. Either it is available later, like for example in case of plants, or you should always have good amount in stock. Then after collecting the sample you have to confirm that the species that you are handling is the same species identification. So we'll look at like what markers and how it is confirmed, okay. And then basically there are two parts, RNA and DNA extraction, that we have to do to go for the molecular biology experiments and sequencing, okay. And then we go for hybrid sequencing, as we discussed earlier also, generating long reads and short reads, link reads, short reads and long reads, okay. And their hybrid assembly followed by scaffolding polishing that we saw in the last lecture, going to final genome assembly and followed by the genome annotation steps. So all these steps we will cover, but today we will look at mainly the experimental part, okay. So basically the sample that has been collected, okay, from which you have to extract the DNA and RNA, right. And particularly for the DNA, to confirm that it is the same species, we will go for the marker gene identification, okay, amplification and confirmation that this is the same species, okay. And then basically the DNA is purified, washed and quantitated, and then you can go for basically the first of all the species identity by using the Sanger method, okay, and later on using other next gen sequencing technologies and third generation sequencing technologies to generate the data, okay. So basically these are the main steps: sample collection, species identification, RNA extraction, DNA extraction, okay, and sequencing of transcriptome and genome, okay. So again the same steps. So now we are dealing in a little more detail. So sample collection is the first step that we will see in the next slide, okay. Then basically the DNA extraction can be done either by using a conventional method, or you can use a specialized kit that is available in your lab for that particular type of tissue, because it will vary from tissue to tissue, plant, animals, insects, or and even the type of tissue that you are handling, okay. The kit will differ, bacterial different kit, okay. Then species identification, basically there is a set of markers that are more reliable and commonly used to identify the species, and for plants, for animals, right? And we will discuss those in later. Okay, then basically again for RNA extraction, triazole method and kit based methods are the two methods of extracting the RNA, right? And also like species specific method, because sometimes it is tricky to extract the RNA, and basically with RNA you have to be more careful because you should immediately process it, otherwise it is prone to degradation. Okay, and then the DNA extraction and high molecular weight DNA is often needed for the long read sequencing, and basically it is also good for the short read sequencing, and also for the high C, which is helps you to construct the complete chromosomal level assemblies, right? And like followed by the appropriate sequencing method. Okay, so for example, samples could be any. It could be a bird, it could be a root, okay, turmeric or rhizome, right? It could be a plant leaf, okay. It could be root or leaf, okay. It could be a very succulent part of leaf, like such as in case of aloe vera, okay, or leaf in general. So basically, it could be plant, animal, okay. So you can collect it from a nursery because there you will at least know, and some expertise is available to identify the species. You can also go to the wild, okay, and based on the morphological phenotypes of that has been described in books, and some expert that accompany you, you can get the samples, because there is a lot of biodiversity in the northeast regions, in the Kerala region, Western Ghats, which is yet unexplored, right? But there you have to start from scratch. You have to look for new species if you are looking for, and morphologically first confirm, and then go for the molecular level confirmation, okay, from any natural habitat, right? Okay, and basically there also, like the sample that you have got should be in plenty, so that you can keep it for later use, after doing the initial DNA RNA extraction and completing your work. Even after the manuscript is submitted, you should have some sample kept with you, stored with you in appropriate conditions, so that you can use it later for any kind of validation or reconfirmation, or any other method that you want to use in the future. Okay, so now the genome that you're interested in, you should have some idea of their genome size. Okay, like aloe vera is 16 GB, turmeric is 1.3 GB, right? That you get from literature. Somebody might have published a paper on that. Or one way is to go for the plant C-value database, that gives you a rough estimate of mostly like a lot of species, their estimated genome size. Okay, then chromosome number, again a plant C-value database or literature studies will tell you, right? And it is not that what you will actually get will match the plant C-value database, right, or the literature reports, because those have used indirect methods to basically know the genome size or estimate the genome size or number of chromosomes. But what you are doing is a direct method sequencing and basically reconstructing the genome, so your values will be more reliable. Okay, although if you find a match between the two, that is a perfect situation. Otherwise you should be ready to defend that the value that you have got is fine. Okay, and of course the ploidy level, because that will be helpful in estimating how much coverage of the genome you should do by sequencing, and also to handle the later complications in the analysis part. Okay, so first of all the DNA has to be extracted, for which there is a conventional method, okay, which like basically you should be having a molecular biology team that is doing this all the time, and or basically there are some DNA extraction kits available where the protocol is defined and you just follow the steps and try to get the DNA. Or you can always optimize the steps, okay, such as incubation period, because you have to break open the cell, so okay, and get the nucleus or without nucleus, and RNase treatment, proteinase treatment. So all these things basically you have to optimize. Okay, and so basically whatever tissue you are taking, you should grind, lyse and precipitate, okay, then centrifuge, adding ethanol, washing, and then eluting the DNA. Okay, we will discuss these in detail later. And basically what the DNA that you have got, you have to purify it and also quantify it before you use it further. Okay, then species identification. So DNA you have got. So now DNA will have the basically the genetic material, right? In that there are markers also present. Okay, so now markers could be present on the in the nuclear genes. It could be around the nuclear gene, or it could be the chloroplast gene in case of plants, or it could be the mitochondrial gene in case of plants or animals. Okay, so basically what marker you are going to use will matter. Okay, and a combination of markers is like even more preferred, that you confirm by using multiple markers that the species that you are saying that okay, this is the species is the same species, right? Okay, so basically for animal it is the mitochondrial cytochrome C gene, oxidase 1 COI locus. Okay, or the other mitochondrial genes such as cytochrome B, 12S ribosomal RNA or 16S ribosomal RNA are used. Mitochondrial is preferred because basically it gives a kind of correct estimate of like what species you are dealing with, right? And also because these mitochondrial genes also do not have introns. Okay, so that also like simplify the problem. Okay, right, and they are haploid mode of inheritance and they are limited recombination, so that is helpful. And in case of plants, basically METK is one gene, maturase K. Okay, and you could also use the chloroplast genes, right? And the other genes, ITS is helpful sometimes and sometimes not. So ITS could be one of the marker. Okay, okay, so ribosomal basically it is in between the different ribosomal subunits, right? So METK, RBCL, tRNH genes are generally preferred. Okay, so like these are the genes like RBCL, which is ribulose 1,5-bis phosphate carboxylase, METK, maturase K. Okay, tRNH, PSBA, which is intergenic spacer region gene, or internal transcript spacer, so which are these regions. Okay, so basically these are sometimes not helpful in case of say angiosperms. The variability between these regions is very low, the 18S and ITS region. So a combination of ITS, 18S and the other these genes, marker genes, is important. So basically it is better to first know that what genes you are going to amplify or standardize this for your lab, right, or for your work, and then do all the confirmations based on that. And of course when you have basically the gene that you are interested in, you have to amplify that gene. So like for example here, so you should have the primers ready to amplify that gene, because the product of that gene you will be sequencing by the Sanger sequencing method. Okay, because it is good to cover like 800 base pair in one side, and almost like if you do forward and reverse, it will be like almost 1500 base pairs, which gives you a good idea about the region that you are dealing with, and also with the alignment with the database, you can be more sure that both the forward and reverse have aligned. Okay, so basically the marker gene amplification, and then you have to do the various molecular biology steps like checking the amplified products on the agarose gel, purification of the product, because or basically you can give the DNA, and this purification part also can be done at the sequencing team end. They also do before this. Okay, and with the primers the Sanger sequencing is done, and when you have got the sequence, for example isolation of DNA and like forward reverse primer, okay, PCR amplification and followed by DNA sequencing. So the DNA sequence that you have got, okay, you can check the quality of that before proceeding further. So it should be good quality and long enough region, so that you can be sure that you are dealing with the right, it will not be failed because of like short regions, and it will not be like an incomplete match. And then you match it against a database of known species which you will find at NCBI. Okay, and you will blast these markers against the NPTEL database, blast 10, right? And if your match is the first match, right, and also showing all the identity, similarity, and coverage and e value, okay, which should be as less as possible, like 10 to the power minus 15 or even lower, okay. So, and it should also be the first match in most of the cases, right? So then you are sure that the species is fine, okay. So this is like just to depict that how the marker gene amplification happens. So basically, right, during the PCR. Okay, so basically I am skipping this part. It is just for information that basically PCR has to be done because you need appropriate amount of the marker genes to go for the Sanger sequencing. Okay, then whatever products you have got, you have to assess that using gel, that basically the amplicon size or the molecular weight of that should be as expected, okay. So basically you will do a the PCR products that you have, you will check on the gel, right, and compare it with the known ladder of fragments, and basically that will tell that, for example, here you are seeing the different markers and they are you can see their ladders, and which tells that okay, you have got the correct gene, basically you've got the correct size, and this is the gene that you are looking for, okay. And there are no random amplification from any part of the DNA, because you might be thinking that you have done the species identification or basically amplified the desired gene, but it could have matched somewhere else and it could be an ambiguous product, okay. Then basically the amplicon that you have got, you have to purify that, and using basically either there are so many purification kits available, okay, and the one of the things that is used is by in vitrogen, okay. And basically the purified DNA that you have got is taken further for the Sanger sequencing or the Illumina or long read, short read, or basically these sequencing, okay. So Sanger sequencing is basically the gold standard for the species confirmation, because you would not like to go for a lot of coverage or depth of sequencing, and also you are dealing with a single gene or product at one point of time, although you may have multiple markers, but you will do them one by one, okay. So Sanger can give you the desired result in like less than 1000 rupees. You can get both forward and reverse reactions done. Generally like 350 or 400 rupees is the cost, right, for one reaction. So basically within that value you will be able to confirm that the species that you're the marker that you have amplified is the same gene and also matches the desired sequence in the database, okay. So basically Sanger sequencing is also preferred because it has low error rate and good enough length, and both forward and reverse primer should be there because it gives more confidence also when you are doing the species identification, okay. So like this is the result, for example, for the METK gene for like Tinospora cordifolia, it is giving the best hit, okay. So basically the amplified product was as forward and reverse were concatenated, and basically you can see that it is giving like the maximum score and total score are same, which okay, and it is very high, and the e value is 0, that means there is no chance of ambiguity, okay. E value as low as possible is better, and 0 is the perfect, okay. It match with your desired sequence by 99%, okay. So basically it will always be that the region that you are interested in or the gene that you are interested in, somebody, for the markers at least, somebody would have put some sequence in the database in the past. Otherwise at least you can get some of the like nearest hit to the same genus or family, okay. And then you have, along with the morphological confirmation or basically or some of the marker, if you can get the correct hit or the desired hit, right, you can confirm that the species is correct, okay. But mostly it should be that one of the marker sequence should be there in the database, already done by someone, submitted by someone, and you can confirm by aligning against that. So NCBI that way is important, and NCBI blast is usually done for this purpose. Okay. Now from where to extract the DNA? So basically depends upon the species that you are doing. For example, plants, leaves is one thing that you would like to consider, because it is soft tissue and not like the stem or root. Okay. And root, basically getting the root is also a difficult task, because for a tree it is very difficult to dig that deep, right, to see the young root hairs. Okay. And basically that may also damage or kill the plant. Okay. So leaf is one of the easiest sample. Similarly for animals, blood is one of the easiest sample. Okay. So leaves are taken, but leaves may have some issue, for example presence of polyphenols or terpenoids, because in plants, basically medicinal plants or aged plant, or basically the so basically young leaves is even more preferred, which have lesser accumulation of these kind of polyphenols or terpenoids. Okay. And okay, so those are used for extracting the DNA and also the RNA, right? So okay, and also it is not that all the genes that you are dealing with will be expressing in the leaves. So some will be expressing in the leaves, some in the roots, some in the stem and other parts. So when you are dealing with transcriptome, as many tissues you can do is is better. But since like across the tissue the DNA is same, so basically for DNA extraction, leaves is one of the good options. Otherwise for RNA it is more number of tissues is better, but leaves is still like good enough to go for the first sequencing project. Okay. So leaf, and it has less of phytochemicals, because it may have flavonoid, terpenoids, polyphenols, etc., but that will be much lesser than the stem or other parts of the plant. Okay. And young tissue is preferred, and which have higher DNA content and lesser of these phytochemicals. Okay. And the goal should be that you should extract high molecular weight DNA, because in the long read sequencing you desire longer products. So if the DNA is already fragmented, so even if the technology is promising and you can give you longer DNA sequencing as a result, if your initial DNA is fragmented, so it will not be good. Okay. So high molecular and high quality DNA both are required. High quality also, because if it is full of impurities, it will block the nanopore pores. Okay. Or basically interfere with the sequencing in the sequencing enzymes or process. Okay. And basically other additives are needed as the depending upon the tissue, and followed by the DNA washing and purification steps. Okay. And once you have done the extraction, washing, purification, you should quantify the DNA. Okay, that how much DNA you have in hand. So you should have surplus DNA, so that your sequencing job is finished, your species identification sequencing these things are done, other molecular biology experiments are done, but still you should have sufficient DNA in stock. Okay. Stored properly, so that you can use that later. Okay. And this also remains as an evidence of the DNA that you have extracted, that it is for future validation, that will be very much needed. So basically for that, qubit is one of the instrument to quantify the DNA, and also the nanodrop. Okay. And basically there are high sensitive and broad range kits which are available. Okay. And also for single stranded DNA, right. So basically they can highly sensitive kit can do up to 0.2 to 100 nanogram DNA concentration it can measure, so which is like very less, and broad sensitivity from 2 to 1000 nanograms. Okay. So basically up to 1 microgram it can assess. So these things will matter, because sometimes the DNA could be in plenty and sometimes it could be scarce. So you should use the appropriate kit. Okay. So qubit is one of the instrument that you would like to have, or for the DNA quantification, because when you are sending the DNA out also for sequencing some agency, or even for your internal validations and library preparation steps, qubit is one of the instrument that will be desired to accurately estimate the amount of DNA. Okay. And the quality is assessed mainly by the this nanodrop spectrophotometer. Okay. Where the recommended ratios of 260 by 280 is about 1.8, and 260 by 230 is 2 to 2.2. Okay, slight variation in this is okay, but if it is too varied, so you will definitely see some errors, or so basically some impurities, or some issues with the later on sequencing. Okay, so basically this DA, basically 260, 280 and 260, 230 ratios are very important to assess the quality of the DNA, to assess the basically impurities because of RNA and protein. Okay, and whatever you do, there could be always an issue with the secondary metabolites. Okay, so plants, that is why, are like sometimes difficult samples, and similarly insects are also difficult samples because there will be some secondary metabolites or cell wall contamination. So these kind of things, okay, may be one of the issues, okay, that will definitely impact your either polyphenols or presence of proteins or RNA, okay, or polysaccharide, impact your 260 280 values. Okay, so basically that is why optimization of the method of DNA extraction and purification is very important for certain tissues. Okay, so you might be using a method that works well for a standard tissue like okay, but for different type of tissues from different plants, animals, okay, you may have, or birds particularly, they are also troublesome sometimes. Okay, you have to optimize the method according to the species. Okay, then genome is one thing, and then transcriptome tells you many things about the genome. Okay, one is the genes that are transcribed at that point of time. That means you have got the RNA transcript evidence for the expression of those genes. Okay, so that will vary from time point to time point. Okay, tissue to tissue, developmental stages, right, even different times of the day. Okay, so basically there will be some variation during various these things. Okay, so but generally for the genomic projects, any one time point but basically as soon as you get the leaves or this material, you should go ahead with the extracting the transcriptome, okay, RNA, total RNA, because if you delay this, it will lead to automatic degradation of the RNA. So basically it should be done as early as possible. Okay, so basically RNA transcriptomics tells you the number of transcripts, okay, type of transcripts, right, and also an evidence for the presence of those genes in the genome, which will also help as a empirical evidence when you're doing the maker or some other gene prediction pipeline. Okay, and also in the scaffolding process in the assembly. So transcriptome is should be done with all the genomes, and even only the transcriptome is sufficient to write a paper, because that will tell you okay, what genes are there, right, and what is their then you can go for the function level annotation of those transcripts, and that will give you a fair idea of the pathways and other genes which are present in that organism. Okay, so basically for doing the transcriptome, also for example appropriate tissue should be taken. For example, leaf is one of the tissue and should be processed immediately. Okay, and then using the appropriate buffer, like it could be triazole or any other conventional buffer or a kit. Okay, RNA is extracted, purified and washed. Okay, then eluted in a nucleus free water, right, because you don't want the degradation, right, and it is, if required, it is also column purified. Okay, and then basically the ribosomal RNA or the structural RNA, because you are right now interested in only the mRNA, right, the protein coding genes transcripts. Okay, so and basically in the transcriptome like 95% or more could be the structural RNA, which could be ribosomal RNA or tRNA. So those have to be removed by ribodepletion steps. Okay, and only the mRNA should be taken for the library preparation. Otherwise you will end up doing 90% sequencing of those structural genes, ribosomal RNA, and your main mRNA will be very less, right. Similarly for microRNA also there are like different steps. Okay, then you go for library preparation and sequencing. Okay, and in between there should be one more step of converting the mRNA into the cDNA, because generally like it is the DNA that you sequence in the sequencer. Okay. Okay, so basically plant tissue is taken in immediately. Okay, I think these steps are similar to previous slide. So basically the idea is that in case of RNA, the tissue should be processed immediately, otherwise you will end up losing a large part of the transcripts and you will get degraded transcripts, and that will not be appropriate for long read sequencing, and even for short read, it will give you you will not be able to reconstruct the transcripts from that shorter sequences. Okay. So for the transcriptome analysis, which is a critical and important part of any genome sequencing project and genome analysis part. Okay, so the appropriate tissue from which you will extract the RNA. Okay, because you want the mRNA, so poly-A purification, and mRNA purification steps are important. Then mRNA is fragmented, reverse transcribed into cDNA, because generally it is the DNA that you are basically sequencing. Okay, adapters are ligated in case of Illumina, for example, and others also, and basically clonally amplified on the flow cell of Illumina, followed by the high throughput sequencing. So generally for one genome, about 15 to 25 GB of transcriptome data is sufficient to cover the entire diversity of the transcripts which are there in that, and also to recover a good constructed transcriptome. Okay, if you have more budget, you can go for higher also, but 15 to 25 GB is like good enough transcriptome from a tissue. Even with 5 GB you can do something, but like we have seen that when there is sufficient amount of RNA data, the quality of the reconstructed transcripts and the information that we get about the genes from that was better, and that also helps in sequencing. Okay, and basically it is followed by the pre-processing of transcriptome reads, like adapter removal, and low quality sequences are removed. Okay, then Trinity is used for de novo construction of the assemblies, of the transcriptome assemblies. Okay, and mapping of raw reads to the transcriptome assembly is done by HiSet2. Okay, and then basically the redundancy in the transcripts removed by the seed heat EST. Okay, and then followed by the gene prediction using transdecoder. These things you will cover in detail later. Okay, and then the transcriptome assembly is used for the transcripts, as well as the transcriptome assembly is used for the constructing the final genome assembly. Okay, so basically we have also discussed this earlier. So transcriptomics is good to study the genes at the transcriptome level, that they were actually expressing and not pseudo genes, and you get a direct evidence of that, and also their respective amounts, and also information about the alternative splicing that might be happening over there. Also, like unknown genes can be known by this way, which microarray and other methods cannot tell, because here you are doing or this study in an unbiased manner. So all the transcripts will be sequenced. Okay, different expression level paralogous homologous genes, because the copy number of particular transcripts, and okay, also act as a step in validation or in gene prediction annotation steps, right? And tissue specific condition, if you do multiple time points, you can know, and in different tissues you will get different set of transcripts which are expressing, though there could be some core ones also, like housekeeping and other genes, right? And it will also give you hints about what secondary metabolism pathways are are present in that plant. Okay, and basically like transcriptome is one of the important parts in the genome sequencing project. Okay, so just to brush up the previous things, basically the gene sequencing of either the RNA transcripts or the DNA. Okay, Illumina is one of the preferred technology because it produces high quality reads, although they are short, but these days the assembly methods have also improved. Okay, and they also act as error correctors for the long read sequencing. Okay, and the steps are basically the DNA that is there is amplified, clonally amplified, because you need sufficient amount for each of the fragment to get a signal out of it, followed by sequencing. Okay, right. So DNA and the library preparation and clonal amplification, and then the the sequencing cycles. Okay, depending upon what kit or the flow cell that you are using. Okay, so this is again the summary of that DNA, because here you have to do fragmentation because you are interested in shorter reads, but in case and appropriate fragmentation of that particular size range, and the insert size is very important here. And in case of nanopore, you want high molecular weight DNA because you want to go for as long sequencing product as possible, right? And then basically the adapter ligation and library preparation and the sequencing. And different type of sequences are available generally for these kind of genomic projects, like NextSeq, which can give up to 120 GB of data. Okay, or basically this HiSeq or NovaSeq. Okay, basically if you want to go for the cheapest sequencing and high quality, and okay, so NovaSeq is the best, which can pool multiple samples together, and it will give a very low cost sequencing with high quality. So generally the choice for you should be NovaSeq. Even if it is not present in your lab, you can also give this outsource the sequencing, and it can be done by a vendor for about 800 to 1000 rupees per billion base pairs, okay, per GB, and you can get the data, right? So basically you can estimate how much you want, you can order it, you can pay like some part for the library preparation, about 10,000 rupees, and okay, this data you can use for your analysis. Okay, then linked it sequencing was important. 10x Genomics was offering it, but these days like link read by 10x is stopped. Okay, but basically the concept was that you generate reads from various fragments. Okay, for example, this is different fragments are labeled differently, and the subreads that are generated from all those fragments are also labeled differently. Okay, so basically each long fragment is sequenced separately, okay, with the label, and later on those labels are used to resolve it. Okay, like these are the different fragments, okay, and they are differently labeled. So the smaller sequences that are generated out of it are also labeled with the same type of marker, okay, and later on sequenced together, and because they have different labels and which tell that they are coming out from that DNA fragment, this one or this one, okay, they can be separated later. So link reads provides a good advantage when you are doing the genomic assembly. Okay, and of course nanopore and PacBio are there for the long read sequencing, where the idea is to get as long read as possible, because that is good for transcriptome also, because you can get the complete transcripts, because the average length in these is like almost 10 KB, okay, which is good enough to cover a complete transcript, right? Which gives you idea of also of the complete transcript with the alternate spliced forms, right? So basically you will get their respective transcript as well as their numbers, copies, right? And also for the DNA sequencing, because these generates very long reads, and if after correction for errors by the Illumina reads by the hybrid approach, they can provide they can help to build very long contiguous assemblies and with a very high N50 and other quality values. Okay, and again so many type of nanopore sequencers are available, which are very affordable, like this is MinION, okay, and MinION MK1C. So this is USB based kind of, you can attach it to the system by this, okay, and okay, so like this one will be like 1.5 lakh per run, okay, and this sequencer again you can get for 5 or 6 lakhs, the sequencer as well as the consumables, right? And these are the bigger versions of this Prometheon and the GridION, which are for the big sequencing labs. But for our purpose, for the researcher level, lab level, basically MinION and this MinION MK1C and normal MinION are good devices. Okay, and we also did it earlier, but again just to refresh, long reads help in basically building more contiguous assemblies, okay, which can be corrected by the short read data, and combination of long read short read is very good along with the link reads, okay, and different insert sizes libraries should be used for Illumina also, okay, to cover the genomes better. Okay, so basically also like for example, from one of these device you can get almost 20 to 30 GB of data, which is like if your genome is 1 GB and you're getting like 20 to 30 GB of data, that means 20x or 30x coverage for your genome. That is good enough for a decent assembly, okay, which can be taken up further for the chromosomal level assembly by Hi-C, right? And this is low cost and error rate are high, but that can be taken care by the short read sequencing. Okay, and basically like just to give an example, like in case of aloe vera, which was 16 GB, okay, basically total long read data generated was 123 GB, okay, which is about 7.7x, but it helped to construct a good quality assembly, okay, not the chromosomal level because it's a huge genome and basically very expensive project, but okay, this much or up to 10x is sufficient to cover the genome well, right? And for a smaller genome like 25x should be there, right? And okay, so, but the problem with the nanopore is that because this DNA is passing through pore, so the DNA is not pure and contaminated by polysaccharides or polyphenols, that may interfere with the like sequencing process and block the pores and failure of the runs and wastage of like consumables. So you should be very particular and careful about the DNA quality in case of plants. Okay, so yeah, so basically each genome will have its own complications and issues, okay, and that is why while you are planning for a species, you should know that what challenges you may face. If it is animal, then basically the tissue and right amount of sequencing that you need. In case of plants, it is always like contamination by polysaccharides, polyphenols, etc. may be an issue, right? And in case of insects, again these polysaccharides and polypeptides and proteins could be an issue. So and that is why selection of the DNA extraction to purification to the sequencing methodologies all are critical and important steps while you are handling a genome, right? Okay, so basically just to conclude, you will be first taking the sample, extract the DNA, identify the marker gene to confirm that the species they are dealing is correct, right? Otherwise you end up losing everything that you have invested in that, because you might have end up doing a species that was not correct, and the basically reviewer and publication also ask for this, okay. And after the purification of this, you will be confirming that species by Sanger, right? And okay, and also like the marker sequences that you are amplifying, if it is not known earlier, that is also one of the contribution from your side and should go as a part of the manuscript and submit it to the journal, right, and NCBI also, okay. And then the transcriptome sequencing, DNA sequencing by both long read and short read, and followed up by the chromosomal level assembly by Hi-C, okay. So that chromosomal part I am not doing now, we will be doing it later. I don't want to confuse too many things now, right? And basically the data requirement, we see like whatever is your genome size, short read data should be about 100 to 200x. So if the genome is 1 GB like in case of turmeric, so at least 100 to 200 GB or billion base pairs you should sequence, okay. Long read should be 20 to 50x, okay. So if their genome size is 1 GB, then almost like 20 to 50x or GB of the short long read data should be there, and link reads about 100x, because that also supplements and complements the Illumina sequence, because ultimately they are generated by Illumina. And for the transcriptome, minimum 15 to 25 GB per sample you should generate, okay. to construct a good quality transcriptome and also for good quality gene prediction. Okay, so in this lecture we covered some very important topics such as how to decide the genome and basically the initial criteria for confirming that the genome that this sample that you have collected is correct. Okay, and yeah, the DNA extraction and quantification is a key component and very important for sequencing. Otherwise, impure DNA will always hamper your sequencing efforts and lead to low quality and low quantity of data, right? And RNA extraction and transcriptome are key component, and okay, that should always be the part of the genome sequencing project. Okay, so this is good for today. Thank you for your attention, and see you in the next class. Thank you. Bye.